The animals which have been genetically engineered to contain one or more genes from an exogenous source. Transgenes are integrated into the genome. Transgenes can be transmitted through the germline to progeny, and the first animal after producing transgenic animal is called as founder animal. So, actually, see the conventionally what we used to do, if we want to have a character or property or one kind of uh, uh, the animal behavior or whatever it is. So if you want to have, we used to do conventionally, just mating the different animals and get the progeny with your expected character. So for example, you have a, a cow which is giving a very good nutritious milk, and it is having heavy and flesh, everything is okay for you, and but some kind of viruses, mad cow disease and all, it is very easy to prone to that disease. But you found another trait um, from some other different place, and which is having that is very resistant to that particular kind of disease, but other kind of characters or properties are not there. So what we used to do conventionally, we used to have this meeting of these two and try to get the progeny, the trait which can have both of the characters, but it may not be 100% you will get all the things. While getting that, there is a possibility you will get the progeny with resistant to the particular disease what you are expecting. That means that character you get, but at the same time you may lose other characters. You are having very nice things which you are having already in your the animal or cow. So the randomly you may take so many trials, and you may get your expected character, but at the same time you may lose some of the things. So there is you have to try many times to keep all the things in your next progeny. So that is what the conventional traits what you used to do. But after the transgenic animal and molecular biology development, and where you can find a gene or set of genes which gives that particular kind of character to that particular animal, what you can do, that particular gene you can put into your animal, so that you will not lose any other character which you are already having, your animal is having, and at the same time you can induce or add that particular character to your animal. So that is a main idea, and that is an excellent application of the transgenic animals instead of the conventional methods. DNA from another animal or species is injected into cells and becomes a stable part of its genetic information. A successful transgenic animal expresses the foreign gene only in the desired location within the body. See, this gene of your interest, it always not from the same kind of animal, same kind of species. Sometimes you can extremely a different foreign gene you can introduce from entirely different animal, entirely different species or genus, like even animal genes you can introduce, so that you can expect if you want to produce human protein over there. So you can engineer such a way that you can engineer such a way to express that particular foreign gene in a particular kind of tissues only, particular kind of tissues of animal. For example, if you want to express a protein, a foreign protein, it may not be animal, the cow protein, or it may be cow protein, but you want to express and in the cow, transgenic cow, and get that protein should be secreted from the milk of the cow. So you can genetically engineer such a way that that particular gene promoter, which is specific to the order of the cow, so that you can, when once you introduce into the animal, it may be present all the tissues of the cow, but it will express only a particular tissue that is ordered, and it will secrete it out from the milk. So that is one of the just example I want to I just giving you. You can engineer such a way that you can express your protein of interest a particular kind of tissues also. And sometimes you can manipulate such a way that you can remove your gene of interest in the sense which you don't want to have. So already in the animal it is present, but one of the genes you don't want, that is giving some kind of unpleasant things, and maybe that is the gene can cause some kind of diseases or some kind of unwanted property of the animal. So you can remove it, and sometimes for the experimental purposes you can remove. For example, um, you want to see how the animal behave, or some kind of other kind of diseases it will fight in absence of particular gene during your research. So you can just knock out that particular gene. So that is also kind of a transgenic animal. So actually, in our laboratory, we are doing my students are doing some knockout studies in the bacteria, not in the animal. So animal, it takes a lot of time to get it. So at present, we are doing knockout studies in bacteria. So in order to see after the gene deletion, what is the property of that particular animal, how it behaves. So that link to your research. So research question. So that is the thing. That is also a kind of transgenic animal. Okay, so what I said, you are introducing a foreign gene of your interest to the animal. So how you can introduce and where you introduce. So you cannot introduce into a already grown up animal. So already born and grown up animal, if you do and you want to change the property of that animal, is not so easy. So usually, I know transgenic animal, when you say when you are introducing at the uh, fetus or embryo stage, so that the resulted animal will have all the cells, or most of the cells, are part of the no, the animal will be having the transgene. So the one gene which you are going to introduce is called as transgene. So, so that is what you will do. But there are other um cases where you can introduce a transgene in an adult animal, so that you can add a property to the particular adult animal. So that one we call as not transgenic animal, is a gene therapy. So certain kinds of abnormalities or to fight some kind of diseases. So due to the once we understand the particular uh, disorder due to some kind of deletion in a gene or something. So that particular gene, if you introduce to the adult animal, especially the human, it may no, it may solve the issues. So what you can do, you can introduce that particular disease, sorry, the gene into particular tissues, like a thyroid tissue you want to target, and you want to introduce that thyroxine producing gene, and using some kind of vectors, usually the viral vectors, and it will introduce the genes into the particular tissues, and it may produce the thyroxine hormone. So that in that case, that is called as the gene therapy, but we are talking about the transgenes where we introduce at the early stages of the um, animal. So like either fetus stage or embryos, different embryonic stages of the animal, so that you can expect the transgene will be incorporated into the all the cells um, of the to be born animal, or some of the techniques may introduce only few tissues of the or few cells or tissues of the animal progeny. Then it is going to be also transgene, but it is not fully transgenic animal. It's a partial tissues will be there. See, procedure is basically the same regardless of the which animal is involved. Some kind of procedure will be different, but the mere methodology, the technique will be the same, but a slight variations will be there based on the different animal. Integration usually occurs prior to the DNA replication in a fertilized oocyte. So if you start at the fertilized oocytes, that means the developmental biology after the mating and after the um formation of the zygote, that means after the sperm cell and the oocyte together form a fertilized oocyte, and where it is a single cell at that time. At the time, if you introduce anything, any transgene, and once you introduce the transgene, and it need to be integrated into the genome. So that piece of DNA should be enter into the nucleus, and inside the nucleus it need to be integrated into the host genome, host genome in animal genome. So once it successfully done, then that particular cell will be having a transgene, and a single cell will be divided into 2 and 4 and 8, and then it will form a mass of 64. Then is a morula, then it is a blastocyst, and after that a few other embryonic stages, and it will produce the uh, fetus. Then it is um, uh, the progeny animal will be, all the cells will be having the transgene. Okay, so majority of the transgenic animals carry the gene in all of their cells, including the germ cells. Transmission to next generation requires germ cell integration, germline integration. I'll tell this statement later. Some integration events occur subsequent to DNA replication, giving rise to mosaic animal, which may or may not contain the transgene in the in its germline. See, if you are introducing your gene of interest instead of when it is a single cell, when you are introducing when it forms like a cell mass, like a morula, either 64 cells or 128 cells or 32 cells, whatever, of not a single cell. After that, you introduce. So what happens? The when you introduce, all the cells may not get integrated the transgene, partially, 50% or whatever the percent it is, doesn't matter. What I'm trying to say, all the cells may not integrate. Let's say what happened. Some cells got integrated, some cells not got integrated. And it will have the three layers of the body, and then it will produce a fetus. Then and the born animal may not have all the tissues transgene. Some tissues will have the transgene, some tissues will not have the transgene. So this kind of animal we call as mosaic animal. So some tissues will have transgene and some tissues will not have transgene. If you are introducing your gene of interest after the division of the fertilized oocytes, that means when it is a single cell, of course, whether it will integrate or not integrate. So it will result into either transgene or not transgene when you introduce in the single, when it is a single cell. But when you introduce after the single cell, after the division, it may result into no not transgenic. It may result into fully transgenic, or most of the times it may it result into mosaic animal. Okay, the mosaic animals also two types. What are those? See, as I told you, mosaic animal is the one animal is having certain tissues are having the transgene and some tissues are not having the transgene. So randomly, those tissues like germ cell lines. So like the male is a testes and other female is uh, the mm, the embryos which are having the transgene. What happens? The next generation, the next generation, it will have the transgene and it will produce a fully transgenic animal. Even though the mosaic animal, which is having the transgene in the germ cell line, mosaic animal is the partially, so some of the tissues are having the transgene, some of the tissues not having. But the tissues, the germ cell line is having the transgene. So the next generation from this mosaic animal, the next generation, what happens? All the tissues will be having the transgene because the germ cell line is having the transgene. So that's why the second generation will have fully transgenic animal, even though the first generation is the mosaic, but that mosaic should have the transgene in the germ cell line. So that is the point here. So how are transgenic animals produced and how do transgenic animals contribute to the human welfare? So the next two classes, this is the things we are going to understand. How it will be produced, that is one of the interesting and need to be understand, and how it contributing to the human welfare. So these two topics we are going to cover. So again, a little bit molecular biology. So what actually the gene of your interest should have? So target gene, the transgene will have its own promoter and enhancers. If you see the eukaryotic genome and human genomes, human genes, it's not only the promoters, a lot of enhancers will be there. A lot of different kinds of promoters will be there. A tightly regulated process, this transcription and translation. So that is how, you know, some of your tissues of, I mean, our tissues. So all the tissues, all the cells are having same amount of DNA, same number of genes. You agree or not? All the of our tissues are having same amount of DNA. I mean, within our body, all the cells, our cells, trillions of cells, will be having the same amount of DNA, same genes. Of course, one to one. So it may differ from you and me, a slight some gene or some DNA, but within myself or within yourself, all the cells will be having the same DNA and the same genes. But each cell is, I mean, there are different kinds of cells, different types of cells, and it will produce different kinds of proteins. Of course, the some of the proteins which are commonly needed will be producing all the tissues, but some of the proteins are expressing specifically in some tissues. Some genes are proteins which are producing in liver may not produce in heart. Some of the proteins which are producing in your heart may not produce in your brain. How it happens? Though all the genes are present, the genes which are expressing in liver, it is present in the brain, but why it is not expressing? So there are some tight regulators at the promoters and enhancer regions, and epigenetic modifications will occur well in the developmental stages. It is not revert back. So those kind of modifications will lead to the those genes will be get silenced or it will keep expressing. So those promoters. So as I told you, if you want to express the particular gene of your interest in the order, so you can engineer such a way that the promoter which are specific to the order, and you can put it that one, so that you can target your gene of interest to produce a particular kind of tissues. Or if you don't want to target any particular kind of tissues, you can put a promoter which can produce in all the tissues, specific to all, not specific to any kind of tissues. So that is the thing. And coding sequence will be there. That is, you know, your codons producing protein. So that is open reading frame. And then polyatyl. In eukaryotes, you will have the 3' modifications. That is three polyatyl. It must be there. So these are all you need to keep your target gene. And gene transfer methods are many. Direct microinjection, virus-mediated gene transfer, embryonic stem cells, nuclear transfers, artificial chromosomes for gene transfer. Okay, and here I'll explain three: direct microinjection, viral mediated, and embryonic. These two we are not covering. So we'll first start with the viral mediated gene transfer. This is the earliest method for successful gene transfer in mammals. Virus has transfection property. As you have seen the bacteria and bacteriophage. So bacteriophage also we are using for the introducing a gene of interest into the bacteria. The similar way, you can take an animal and the animal virus also you can use engineer the animal virus such a way that it will inject its genome into the animal cells and it will get integrated. And it's a natural property as bacteriophage what we understood earlier. Here the virus introducing a gene of interest, your gene of interest, is a natural property. If you make the virus, so the time consuming part is you have to engineer the viral genome such a way that it carries your gene of interest. That is the time consuming thing. The same thing in the bacteriophage also. The time consuming part is you have to introduce your gene of interest into the bacteriophage. So otherwise, the efficiency is very high. So once you introduce this virus and introduce to the cells where you want to add your transgene, is a natural phenomena. All the virus it can introduce infect the target cells and inject the genome into the cell, and also is a natural phenomena. The bacteria, the viral genome, once it gets introduced into the cytoplasm, it will get into towards the target to the nucleus. This is also natural and go and integrated into the genome. So that is the main idea, actually utilizing this virus as a of um, mode of introducing the gene of interest. Killed virus is replicate defective, and the virus gene is replicated with transgene, and transgene is delivered to the host cell by transfection. So if you do the introduce the transgene in the adult animal, we call as a gene therapy, can be used to transfect the wide range of cells, embryonic stem cells or embryo cells, or even adult cells. So when you put adults, then it is called as a gene therapy in the animal itself. If you do this, and we are talking about the transgenic animal, so we need to use either embryonic stem cells or embryo cells. So this is the diagram which will explain you to visualize what I'm actually trying to explain to you. So let's say this is the mice, so mouse. This one is having the pregnancy and it is having an 8-cell embryo, and it is there already inside. So or you can separate it out, and after separating out, you can take this virus, this genetically engineered virus, which is carrying a transgene of your interest, and you transfect that virus with this embryo. And after that, as I told you, if it is not one single cell, if you are introducing at later stages of the single cell, that mean 2 or 4 or 8, and then it may leads to the partial, I mean partial in the sense, part of the cells will be integrated. See this, the difference. See, there are three. um it is leading to the three fetus. So what happens? Um, the some of them, as one of the, see this mouse after its cell embryo, it may again its cell may lead to the entirely different embryo. So that is what can happen. So when you introduce this one, either into the all the 8 cells, or some of the embryos it may take and some of the embryos not take. So either I mean whatever the thing, it may lead to the different kinds of embryos. Some embryos will having the transgene, some embryo will not have the transgene. So the resulted progeny, and the one which is having the transgene is called as a transgenic founder. That's a founder animal. And other few which are not having the transgene will be considered not as not a transgenic founder. So how you can find whether transgene is present in the genome or not? So there are some techniques are there, so which you are going to learn in the second part of the genetic engineering. One of the method is southern blotting. Southern blotting is that, so if you want to find a particular gene is present or not, or particular DNA is present or not in your sample, so you can use a probe which is antisense to the gene, and using either radioactive or different kinds of the fluorescence techniques, you can do the detect the gene of interest or a DNA of interest present or not by southern blot techniques. So that is one of the way you can detect, and you can find out, okay, this mouse is having the transgene. So you don't need to kill the whole the animal. You can take a part of it, like usually the scientists or we do, we cut the a part of the tail and isolate the genomic DNA from it and do the southern blot. As well as another technique you do, that is also you are going to learn in the second part of the genetic engineering, that is PCR. So you once you know the gene of interest sequence, so you can design the primers, forward primer and reverse primer, and extract the genomic DNA from the progeny founder animals and do the PCR. So try to amplify using the primers, whether it get amplified product or not. So amplified product you will run on the agarose gel electrophoresis to find whether the DNA is present or not. So agarose gel electrophoresis you are going to learn as one of the practical I'm going to teach you. So once you come back, so when you run and see the gene of interest is present or not. So thus you can understand whether the transgene is present or not. So once you find the transgene is present and found that transgenic founder, then you go for the second generation where you will get a fully a transgenic animal. So that is the viral mediated. And the second method is direct micro injection. So here what you do, you have to collect this um, after the mating, you are going to collect the fertilized oocytes. So that fertilized oocytes we are going to use to inject the DNA molecules directly into the male pronucleus. So I'll tell you what is male pronucleus and female pronucleus in the next slide, where you can see the picture. Most popular technology commercially available, and the success rate is 10 to 30% depending on the skills and the constructs. The efficiency is not related to the copies of the transgen genes injected. So these are all points I'll explain to you. So I want to show you. Yeah, this is the thing. See, after the mating of the female and male, and what you need to do, you have to collect. Of course, you have to sacrifice that animal and take the oviduct, and where it will be having a lot of fertilized oocytes. You have to take that all the fertilized oocytes, and it will be appear like this, and under the highly magnified microscope you can see like this. And this fertilized oocyte is having the, even though it's a single cell, it is having the two nucleus. This two nucleus is after immediate mating, after immediate fertilization. It will take a few hours of time for the two nucleus to be get integrated. It takes time. So during that step you are collecting. So that means it is having the male pronucleus, which is the larger one, and the smaller one is the female pronucleus. So after mixing together, it may become zygote. So that stage you are taking, and after the taking, you take into the petri plates and take this. This is the instrument of the DNA micro injection apparatus, where it will have a small this kind of production, and where it will have the vacuum which will hold the cell. Okay, and this is the micro needle, and the needle is connected to the syringe, where you will have the DNA of your interest. And then this needle will be, everything will be seen. You cannot see directly with your with our naked eye. So everything we will be doing, we will be handling with our hands, but we will be observing in everything under the, everything we will be observing under the microscope, on the screen. Actually, it will be connected to the big screen, and we will be observing and moving everything, the needle such a way that it will poke the cell, and poke the male pronucleus. Once this needle enters into the male pronucleus, then you can release the DNA. There will be some buttons where you can just release your DNA of interest into it, and then you can remove back. That is also just buttons and screws will be there, just operate. So it's a very big mission. So actually, I was operating this one during my PhD when I was in Germany. So it was a tedious job. It will take long hours to do this. You need to have a lot of patience, a lot of handling experience. So at the just a single time you will not be expert. It takes a lot of time to be get expert, and you can able to handle to introduce. That is one of the drawback. So it's one is a very expensive mission, and it required really experienced expert to do this one. And another limitation is, after you doing this one, say the oocytes you are collecting and doing, if you start with the introducing into 30 or 40, all the cells may not survive. So because you are physically, you are poking it, and may not be survive. It may blast, and it may die. And from 30 it may end up with that 15 or something, 15 or 20. Some of the times just it will die. And after that, what you need to do, these cells, this fertilized oocytes, after introducing the foreign gene of your interest, Hello, am I audible to you?
Yes, sir.
Oh, doing recording. Okay. So that is the idea. So so male pronucleus you are preferring because that is the larger in size. That is the main reason, and easy to observe and easy to inject it. And after that, this fertilized oocytes you are going to introduce into a different uh, female mouse, and you are going to introduce for the implantation. So that different mouse, any different female, it may not be going to implant immediately. So for that, you need to do something here. So let me start from the picture again from here. So you have to make the male and female, and then collect the fertilized oocytes, and the DNA you are going to introduce into the male pronucleus under the microscope. And this is the called as direct microinjection. And after that, this one you are going to introduce into the pseudo pregnant females. What does it mean? So you have to again a different set of mouse. You have to collect it, and you have to take the sterile male and normal female, and you have to make these two together so that it will make the environment of the embryo, sorry, not the, yeah, the uterus, such a way that it will be ready for the implantation. And after this, you have to collect the female and then introduce these fertilized oocytes into this female. And after that, the next progeny, it will leads to the transgenic founder animal. So that is what it will be. So this is another schematic representation, or the figure representation, the same procedure. And after transgenic mouse, you can use a southern blotting to understand there will be so many founders and all the things you run, and you may get southern blot band based on the bands. You can understand which animal is having the your gene of interest. The third method is embryonic stem cell method, where you collect an embryo, especially the blastocyst stage, and this is the blastocyst. Okay, so blastocyst stage, if you collect, it will be having a single layer of single layer outside, and inside there will be mass of the cells and some empty place. So this mass all will be like a um, pluripotent stem cells. So what is a pluripotent stem cells, and how it works, everything maybe we will continue in the next class.